Nucleotide Chemistry. Nucleic acids. Nucleic acids are polymers of nucleotides joined by 3,5-phosphodiester bond. Types. There are two types of nucleic acids: deoxyribonucleic acid and ribonucleic acid. Properties of nucleic acids. Nucleotides are polyfunctional acids. Nucleotides bear a negative charge at physiologic pH. They absorb UV light at a wavelength of 260 nanometers at pH of 7. Aromatic amino acid wavelength is 250 to 290 nanometers. Nicotinamide adenine dinucleotide has a wavelength of 340 nanometers. Porphyrin has a wavelength of 400 nanometers. UV rays are mutagenic. The mutagenic effect of ultraviolet light is due to its absorption of UV light by nucleotides in DNA that result in chemical modifications. Composition of nucleotides. The components of nucleotides are nitrogenous base plus a pentose sugar plus phosphate group. Nitrogenous base. They are nitrogen-containing heterocyclic ring structures. Two types of nitrogenous bases are purine and pyrimidine. Purine bases are adenine and guanine. Minor purine bases are hypoxanthine 6-oxopurine, xanthine 2,6-dioxopurine, uric acid 2,6,8-trioxopurine. Pyrimidine bases are cytosine, uracil, and thymine. Modified nitrogenous bases are dihydrouracil, pseudouridine, 5-methylcytosine, dimethylaminoadenine, 7-methylguanine. Important points. Cytosine is present in both DNA and RNA. Uracil is present only in RNA. Thymine is present only in the DNA. Most common modified bases are methylated forms of major bases. Pentose sugar. There are two types of pentose sugar: D-ribose sugar in RNA, 2-deoxy-D-ribose sugar in DNA. Composition of nucleoside. It is made up of a nitrogenous base plus pentose sugar, C1 of ribose or deoxyribose sugar joined covalently to N1 of pyrimidine or N9 of purine by beta-N-glycosidic linkage. Nucleosides are N-glycosides. Composition of nucleotides. They are made up of nucleoside plus phosphoryl groups. Usually, phosphoryl group is attached to 5-hydroxyl group of pentose sugar by an ester bond. Additional phosphoryl group are attached by an acid anhydride bond to form nucleoside diphosphates and triphosphates. Ribonucleotides and deoxyribonucleotides are as follows. Comparison of DNA and RNA. DNA. It is mostly seen in the nucleus. Pyrimidine bases are thymine and cytosine. Sugar is deoxyribose. Pyrimidines equals purines. They're not destroyed by alkali, usually double-stranded. RNA. It is mostly seen in the cytoplasm. Pyrimidine bases are uracil and cytosine. Sugar is ribose. Pyrimidines do not equal purines. They are destroyed by alkali and it is single-stranded. Important points. Functions of nucleotides and nucleotide derivatives. Nucleotides are building blocks of nucleic acid. Nucleotide derivatives, cyclic AMP and cyclic GMP, are second messengers in hormonal pathways. S-adenosylmethionine is a nucleotide derivative that is an important methyl donor. Adenosine 3-phosphate is an important sulfate donor. Nucleotide derivative in glycogen synthesis is UDP glucose. Nucleotide derivative that conjugates bilirubin is UDP glucuronic acid. Nucleotide derivatives that act as coenzymes are nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate, flavin mononucleotide, and flavin adenine dinucleotide. Flavin mononucleotide has no adenine in its structure.